Welcome to our UW Data Science Seminar. This is our high-profile external seminar speaker series sponsored by the eScience Institute. And today our speaker is Bradley Voytek. Bing Brunton is the host, and I will turn it over to Bing to provide an introduction. If you have questions, please use the Q&A box on the bottom of your Zoom window. Use that instead of chat or participants and raising your hand, because we'll be monitoring the Q&A. All right, thank you.
All right, so it was great pleasure that I introduce our data science seminar speaker for today, Dr. Brad Voytek from UCSD. Ariel Rokem and I invited him to visit last year, and I was really glad that he quickly agreed to visit us in Seattle. And I was equally glad that he was game for turning his visit into a virtual one when the travel became infeasible. Ariel, for those of you who may not know, has a new epsilon in his family, and so I am here trying to hold down the fort. So Brad is an associate professor at the University of California in San Diego, where he is affiliated both with the Department of Cognitive Science and the Halijilou Data Science Institute. He has a bachelor's degree in psychology from USC, University of California. He then did his PhD in neuroscience at UC Berkeley, where he worked with Robert Knight. Then he had postdocs, both at Berkeley and UCSF, with Mark Esposito and Adam Ghazali. And so I think he's been, can I say you're solidly a Californian, Brad? Is that all right to say that?
Yeah, that's pretty fair.
Based on your background, I think that's appropriate right now. So since 2014, he has led a lab at UCSD where his work has been really influential in terms of contributing to our understanding of neural activity, and especially this mysterious and often misunderstood brain waves that we've all heard about that tie together the activity of individual neurons and large-scale brain function. And in addition, his lab has made really pioneer methodological contributions to a bunch of open source toolkits that have enabled a lot of novel analyses and understanding of neural activity, especially relating them to human cognition and disease. One of the reasons that we invited him here for the data science seminar is because he has been a tireless advocate for data science education as well as open science. And I know that the students in my lab have often been really excited to follow his publications when they come out and make use of the tools that they have released. So we're really, really excited that he came to visit. I will share one more thing about Brad before I turn over the floor. And so even before I learned about his research, I first heard about Brad Voytek because he, as some of you may know, is the world's leading expert on the neuroscience of zombies. And so if you've ever wondered about whether zombies dream of undead sheep, you should go ahead and check out his book. So I'll echo what Dave said earlier. Please use the Q&A session, the Q&A window. I'll try to monitor that throughout. You can also upvote each other's questions if there's someone else asks a question and you really want it answered. Brad has put in one break in the middle of his talk, and so we'll take a small break in the middle for some questions and hold the remainder of the questions to the end. So please join me in welcoming Dr. Brad Voytek.
Thank you. I'm going to go ahead and share my screen now. The zombie brain thing, I can't believe I got a job after writing that book. So, you know, I actually could scrap this entire thing and I can give an entire presentation on zombie brains if you all want. I don't know, Bing, what do you think?
I'm game for it. It is the end of the world.
Much more entertaining than actual neuroscience, I feel like. Well, maybe that's not fair. I'm excited about this stuff, but it's like more niche than zombie brains, I feel like. This is my lab in, well, back when we could still all get within 6 feet of each other, earlier this year in San Diego. And I'm going to be talking about a lot of work today. Most of the empirical results I'm going to be showing you are from PhD student Tom Donahue, Matar Haller and Augusta Shiestuk, who are friends of mine from the Berkeley days, and Richard Gao here, and Scott Cole. A lot of new lab members who are contributing to some of the work that I'm talking about. I'll be continuing it. My postdoctoral researcher, Natalie Shvarnikov, and the other postdoc, Stephanie Martin, who is sadly leaving the lab. She got a job at Google X. I mean, I'm very excited and proud of her for that, but I want her to stay. And new PhD student, Sydney Smith, and new software developer, Ryan Hammonds, who I have yet to actually meet in person, hired during quarantine. So I'm very much looking forward to one day meeting Ryan. And then my former postdoc, Eric Peterson. So also support, thank you for these different organizations for letting me continue doing the work that I like to do. And it's kind of funny, I was thinking about like, where did everything start? And that led me to the presentation I'm about to give you today. And so the state of the Voytek lab in 2014, but when I say lab, that makes it more grandiose than it was. It was me and my postdoc, Eric Peterson, and my lab manager at the time, Torben Noto. This is basically it. Oscillations are pretty neat. We are a fan of neural oscillations and are super curious about what do they do, if anything. Part of the reason that I became interested in neural oscillations during my PhD work is it's one of the things that you can record in both humans and animals. So oscillations provide a nice bridge between species. I'm not a single units are the only thing in the system worth studying kind of person, but we can't really study them in humans except for incredibly rare cases. So given that practical limitation, but you can study oscillations in animals and other species, it's a pretty good way of bridging some of the model work that people do with the human cognitive stuff. The funky thing about oscillations is that they're pretty much correlated with anything. There are tens of thousands of papers that have been published on oscillations over the many decades since they were first reported at the advent of EEG in 1929, of the first actual publication of EEG. And the funny thing about that, when I say they're correlated with everything, I actually gave a talk once where I said, they're correlated with everything. I bet you could find like oscillations in chocolate or something like that. And at the end of the talk, somebody raised their hand and they said, I found on PubMed, here's a couple of papers about theta oscillations and chocolate craving. In rodents. So yes, that is apparently a true thing. You can come up with things and you'll find correlations. The work that I was doing towards the end of my PhD and the beginning of my postdoc was looking at oscillations as a potential mechanism for distant brain regions to be able to coordinate information between one another. So there is this phenomenon of phase synchronization. This is a very busy slide intentionally because I'm not going to be talking about this. I just want you to get an idea of like, this is where I started from, like really overthinking oscillations and their potential for acting as a communication system to coordinate distant brain regions. I said we can't record single units from humans. That's not always the case. There are scenarios in which we can record single unit activity. And if we can't get single unit activity, we can still get very high SNR, signal noise, electrophysiological recordings, field potentials from people who are undergoing surgery for epilepsy. So a lot of the work that my lab does is working with these kinds of patients. So for whatever reason, some proportion of the population who has epilepsy has very severe epilepsy, and a small percentage of those people have epilepsy for whatever reason that is not well controlled by anti-epileptic drugs. And so in that proportion of the population, they'll often choose to undergo an invasive brain surgery that removes piece of tissue, the epileptogenic zone, the part of the brain that the seizures seem to be starting from. And the only way we have to identify that part of the brain where the seizures are coming from is to implant the patients with chronic recording electrodes for a week or two while they're monitored in the epilepsy monitoring unit. UW does a lot of this. Kai Miller, who is a friend of mine who did his MD PhD work at UW, really did a lot of work on this. I do the warning slide because I'm about to show what it looks like in the operating room if you haven't seen it. I've had an undergrad pass out on me before, which is why I show this. So if you are prone to lightheadedness when you see blood, now is a time to maybe look away. So this is what it looks like in the operating room. This patient happens to be awake at the time of the surgery. So this is the awake mapping. You can see the little numbers on the brain surface there. Okay, you can look back now. Those little numbers are actually stimulation sites where the surgical team was stimulating while the patient was awake to identify, in that case, I think it was speech and language parts of the brain in that patient to make sure that when they went to remove the part of the tissue or part of the brain, the tissue causing the seizures, that they didn't interfere with speech and language processes. One of the nice things about working with these patients is honestly, they're like, it's a kind of a pleasure to work with them. It provides a very rare opportunity to get data quality that you otherwise cannot get in humans. They're usually just really, really happy to be doing something and to be helping, right? It's like taking advantage of a unfortunate situation to try and help the process of science. And so I really enjoy working with these patients. Did a lot of work looking at oscillations as a potential communication system. But what I want to talk about today is the state of the lab right now, which is we're not even sure what an oscillation is anymore. We've kind of talked ourselves out of understanding even something very basic, which is what do we mean when we say a neural oscillation? And so we've taken a couple of different approaches to try and figure out, first, where do oscillations come from? And so this is a paper that we had last year in Cell Stem Cell working with cortical organoids. And this is with our collaborator, Allison Motry, here at UC San Diego. Allison Motry and a couple of other folks have really pioneered the creation of these 3D cortical organoids. I am not a cell biologist, so when I give this talk, I just say, through the magic of cell biology, what they managed to do is they take skin samples from people, and they can convert those skin samples into pluripotent stem cells. And then once they are stem cells, they can grow them forward as neurons and neural tissue. And these are about 4 millimeters in diameter, about 100,000 neurons or so. About the size of a bee's brain, and these are, little laminated, people call them tiny brains, right? And the work that we're doing with Motry and the way that we ended up collaborating was looking at I attended a talk that Allison Motry had given, showing that these 3D cortical organoids seem to spontaneously laminate. They separate into cortex-like structures like you would see in humans. And that happens slowly over time as these things grow with a variety of different growth factors. And what's kind of neat is you can then do multi-unit recording from these organoids. And when I saw Motry give a talk, you were showing some of these spiking rasters and talking about these cortical organoids as a model for understanding human cortical development. We don't know a whole heck of a lot about early human cortical development because we don't have ways of studying that. And so if you could have a model system to allow you to understand how the cortex develops, that's a pretty powerful, human cortex develops, that's a pretty powerful thing. But when I saw these spiking rasters, I was like, that's not what healthy cortical spiking looks like. And so Motry and I got talking about, you can see over here, 1 neuron spikes in this multi-electrode array. Everything seems to spike within a short time period. Everything goes relatively quiet for tens of seconds, and then you get another sort of network spike where the population tends to spike again. And so I got asking about, it turns out early in development, the main inhibitory neurotransmitter of the brain, GABA, is actually excitatory until it flips sign. As a lot of people know, during development, you have more neurons and more synapses than you'll have in your adult life. So everything is sort of overconnected, and then there's a period of pruning back of these synaptic connections. And so we're kind of curious about, well, why does this kind of phenomenon happen? Is it prior to the GABA sign flip? Is it pre-pruning or what have you? What happens if you let the system, this model kind of go further over the course of more months? And so what we saw is that over the course of many more months in this growth medium, these cortical organoids go from showing this like network spike to relative silence to, this is time locked to just the network spike events. You can see by the time you're at about 6 months, when the network spikes, you have these kind of ringing secondary spikes. And if you look at the local field potential within this population, you start to see the emergence of oscillations. As it develops even further, you start to get differentiation of frequencies. So the oscillations and the sort of post-network spike ringing start to separate into different frequency bands. And so the number of subpeaks after that first network spike quickly increases from 1 subpeak to 4 to 5 subpeaks at around 25 weeks, and then begins to sort of trail back off. I'm actually going to skip this for the purpose of time. And so what we ended up showing in that paper was that we can, that paper was almost descriptive in the sense of it just shows that we can look at these cortical organoids as a potential way of examining the spontaneous natural development of these oscillations. Now there's a lot more work that we're doing in this ongoing collaboration to try and figure out what happened just prior to the emergence of these sort of secondary network spikes in the oscillations. Is it this reversal of GABA? Is it the pruning, some combination thereof, something else we haven't thought about? So that's all ongoing work. That's just a little bit of teaser of one branch of what the lab is doing through collaboration of trying to understand where do these oscillations come from. But like I said at the beginning of this talk, one thing that we're really concerned about is what is an oscillation? And so going back to Biyu He and Yuri Buzhaki, they have pretty simple, straightforward definitions of an oscillation, which is if you take a time series and you look at the power spectrum of that time series, you have some background process or noise, which has a 1/f-like process in the brain, and I'll like unpack that in a second. But then you have bumps atop of that, whatever background process is, and those more narrow band bumps are the oscillation. So just to kind of orient everybody, on the y-axis here is power, on the x-axis is frequency. So I think most people attending this talk probably know, but just in case you don't, any time series, anything that you record over time, voltage, which is what we record in most the brain studies that we do, the neural studies that we do, it could be stock market prices, it could be counting the number of leaves per minute that you see fall off of a tree. Doesn't matter, anything you can count over time at regular intervals, you can decompose or perfectly represent that thing of the number of things over time or the values over time as a sum of sine waves of different frequencies and amplitudes. So high amplitude, low amplitude. And so that's what the x-axis is the amplitude or the power of that signal, and or the y-axis, sorry, and the x-axis is the frequency. So very slow oscillations or very slow sine waves, I should say, at the lowest end here, and exponentially smaller amplitude at the highest end here. And if you don't plot this on a log-log plot, it looks sort of just like this. It's an exponential kind of decay that is described as 1/f or 1 over frequency. When you plot it in log-log, it's a relatively straight line with these bumps on top of it. And those bumps, if this happened to be a bump at 10 hertz right here, that would mean at about 10 cycles per second, alpha oscillation is how most people would call that. You have extra power above and beyond whatever this background thing is. So how do we measure these oscillations? So I'm going to introduce you to a couple of approaches that we've taken in the lab. And so what I'm going to be showing you today, a lot of the software development in the lab has been done by a PhD student, Tom Donahue, and now being aided by Ryan Hammonds, my software developer. We have 3 open source Python packages that you can install: NeuroDSP, FOOF, which is a ridiculous name. That was a joke that stuck. It's fitting oscillations and 1/f, which is where that comes from. And then the bicycle package, which I'll be talking about. The latter 2 here, the NeuroDSP is neural digital signal processing, which is a couple of functions for simulating LFP-like signals. Here's how we traditionally measure oscillations. So you take somebody's brain data or an animal's brain data or what have you. If I'm a visual researcher, I know that as a visual researcher, alpha is my visual cortical rhythm, and alpha is defined as 8 to 12 hertz. So I take my data, I turn it into this frequency domain, and I look at this 8 to 12 hertz window, and I average the amount of power in this window, roughly speaking. That's how we do it. That's traditionally how, and all approaches that we take to doing this are roughly similar. Wavelet-based approaches, Hilbert transform, they all roughly do the same kind of thing with different mechanics under the hood. The problem with that is if I'm a visual researcher and I'm studying alpha during attention or working memory, I know going in from historical records and tons of studies that if I present a visual stimulus to somebody, alpha power will drop. Whether or not you are aware that this is the model that you have in your head, what you are supposing happening to your data is this, that initially you have this thing background here, and then when you present a flashlight at somebody or present a stimulus that they have to attend to, their alpha power drops. This is kind of what you're doing in your head. This is what you propose, you think is happening, whether or not you know it, when you say alpha power decreases during attention and working memory. The problem with this is there are many different ways of getting a reduction in power when you just look at this narrow window slice. So for example, we know that it's possible for oscillations to speed up or slow down. And if you're just looking at this 8 to 12 hertz window, and I'm exaggerating the effect here, but you can imagine if the oscillation slows down, so the whole bump here kind of creeps back, and you're only looking in this window, the amount of power you have in your oscillation, because you're just looking at this narrow window slice, will appear to be decreased. But that's not really the best way to describe what's happened to the data, because the power actually relative to the background hasn't changed. What's happened is the oscillation has slowed down outside of the narrow window that you were examining. Or alternatively, I mentioned Kai Miller earlier. He showed this work along with Roy Mukamel and Jeremy Manning, that the entire power spectral broadband can shift up or down. Whatever that background process is, the whole thing can move up or move down. And again, if you're just looking at this narrow slice, you'd say, aha, here's what my signal was before in the like light background here. And now it looks like this. But if you're just looking at this window, you'll say my alpha power has dropped. But again, that's not the best way to describe what's happened to your signal. And finally, also, Ella Polvani, who's now in BU Huz Lab, Richard Gao from my lab, a number of other folks have shown that the entire power spectrum can also kind of rotate around a apparent pivot point. And this is like a spectral tilt, it's called. And that pivot point's usually between like 20 to 30 hertz or so. And again, if that entire power spectrum rotates from here to sort of flattens out a little bit, if you're just looking at this narrow window, you'll say my alpha power has dropped. But it's only because you're looking at this narrow slice. And this seems like trivial and straightforward, but when I first started talking about this, people were like, yeah, but prove it. And so it turns out you can't just go around telling people, I think you're analyzing your data wrong. If you really want to try and convince people, you have to provide them with tools to analyze their data the way that you think they should be, which is why my lab has accidentally become a methods development lab over the last couple of years. We're really, truly only developing these methods to try and prove the scientific point that we think is really important, which is all of those give the same exact result using standard analysis methods. If you just look at the power in the 8 to 12 hertz range, you will get the exact same numerical result from pre-stimulus to post-stimulus. But all four of those represent different physiological processes that can give rise to the same apparent numeric output. So in other words, we can adjudicate between any of those four and other possibilities, which I haven't even thought of, without careful parameterization. And so the other problem is, like I said, you can take any time series and you can represent that time series as a sum of sine waves. And this is what it looks like, by the way. So here, let's say this is your time series in red. And if you take that time series and you add a bunch of different sine waves of the same amplitude in this case, of 3 different frequencies, slow, medium, fast, and then some very little bit of noise at other frequencies, which is what all these very low amplitude things are around there, those will sum up to give you this red signal. So the way that we decompose our signals to try and identify oscillations is by using this Fourier transform, decomposing our signal into sums of sine waves. The problem is neural oscillations are non-sinusoidal. And so here is a sine wave, but here is the variety of the kinds of rhythms we see in neural data. You get these sort of almost square wave-like things with a little bump at the beginning. You get these sawtooth kinds of waves. You get these triangle types of waves down here in the visual cortex. You get a lot of interesting non-sinusoidal features. And the problem is, like I said, neural data is non-sinusoidal, but the methods that we use assume sinusoidality, and this matters. And it doesn't only matter from like a neural data analysis perspective, but it also can matter in like real world cases. So I'm going to give another example here of a study we did a couple of years ago, not with this patient. This is a different patient, but this is a patient with Parkinson's disease, presenting with very classic bradykinesia, which is a symptom of Parkinson's disease, which is characterized, it literally just means slow moving. And it's, you can see sort of the gentleman with slumped shoulders here. You'll see also a resting tremor in his left hand when I play the video, and very, very tiny steps in slow moving. And so this same gentleman was implanted with a permanent electrical stimulator, deep brain stimulator, into his subthalamic nucleus. And that permanent electrical stimulator is turned on. And when you turn on that deep brain stimulator in that same gentleman, this is the output. Now, what I'm about to say is like scientific, in some ways, trivialities. Like we have a theory about why this deep brain stimulation works based upon the non-sinusoidalities of the neural data. This guy doesn't care. That guy doesn't care about, is it non-sinusoidalities or not non-sinusoidalities that, you know, like at the end of the day, there's like a quality of life that works in the clinical care system. And so, you know, got to kind of ground the neuroscience work that we're doing in the fact that sometimes, even if we don't know how things work exactly, it can still dramatically improve the quality of life. Our job as scientists is to get that like little bit of extra information to figure out how to make it 1% better or 5% better, right? But in the meantime, we're going to keep doing this. Clinicians are going to keep doing this, regardless of whether or not the mechanism by which deep brain stimulation works is altering the non-sinusoidal feature of the beta oscillation in the motor cortex. And so this is work from my PhD student, Scott Cole. Natalie Shornikov here, my postdoc, wasn't involved in this study, but she's really like taken up the banner of non-sinusoidal rhythms in the brain in the lab, and she's got a lot of really cool work in progress right now. But Scott Cole came to me working with his collaborator, UC San Francisco, Phil Starr, and Cora DeHempteen, who had shown previously in a couple of really cool papers that when the deep brain stimulator is turned on and you look at this beta 20 hertz oscillation in the motor cortex, which is pathologically strong, they say, in Parkinson's disease, that when you turn the deep brain stimulator on, high frequency gamma oscillations above 100 hertz decouple from the beta oscillation. There's this really cool phenomenon called phase amplitude coupling, which I was demonstrating in that first complicated slide that I didn't talk about for my work, wherein people argue that you have two different neural oscillatory processes, a low frequency one and a high frequency one, where the low frequency rhythm, the phase, if it's at the peak or at the trough, say, biases the probability of neurons spiking, which is measured as high gamma activity above about 80 to 100 hertz. And so that's like a two process thing. You've got two different processes interacting. And that phase amplitude coupling between these two rhythms is pathologically strong, they say, in Parkinson's disease, and then decreases with deep brain stimulation. And Scott Cole came along and said, I don't think that's what's happening. I think the apparent coupling between a high frequency and low frequency rhythm is a byproduct of the math, what we're asking the Fourier transform to do, which is if you look at the individual beta cycles, they can be very sharp here, like 4 and 6 are individual cycles of this beta oscillation. And when they are sharper, in order to mathematically capture those sharp bits in the peaks of your signal, that's done by having more power at higher and higher frequencies. And so you get these sort of spectral stacks. You can see like the more red here is more power. And when they're sharper, the power is higher. It bleeds up at higher frequencies. And so this apparent phase amplitude coupling is actually arising as the sharpness of the individual peaks of the oscillation. And what he showed is that in deep brain stimulation, what seems to be happening is rather than these two different oscillations being decoupled from one another, the deep brain stimulation is actually just smoothing out the sharpness of the individual beta waveforms. And I'll go into that in a little bit more, but I wanted to take a break here, A, so I can catch my breath and have a drink, but B, to address any questions for clarifying. And I'm just drinking water right now, although that wouldn't be my inclination. So any questions? No? Bing?
I think there's I'm gonna maybe just give everyone a quick minute to think about it, and
okay.
If they're drinking something we can't see, so it's great. We have a question from Nick Steinmetz. Nick, I'm just gonna read it. You define an oscillation as a bump in the power spectrum. Some oscillations have a mechanistic basis for being periodic. When the process takes a large value, there's a force that pushes the value down in a negative feedback way, like a spring. But even processes without this property can have power concentrated in a bump of frequencies, as in a Markov process switching between two states. I think most people would not call this an oscillation. Do you have any suggestions for how to identify a mechanistic oscillation versus other power spectrum could have a bump?
So I do not immediately, but when I show the power spectrum there, we're throwing away half of the information, which is the phase spectrum, right? And so one possible way of addressing your question would also be incorporating phase information. I'm trying to imagine what that would look like in the Markov, two-state Markov process. But you could contrive many different, okay, so just visually, the power spectrum doesn't tell you everything that you need to know, and I'll give a very trivial example that I think is maybe a little bit clearer from the one that you gave, which is if you have two separate oscillations at like 10 hertz and 20 hertz, and those oscillations can be phase synchronized or not, doesn't really matter. There's just like two different ongoing oscillations. you will have, clear, oh, that doesn't work. You'll have two clear bumps in your power spectrum, right? But just as easily, you can imagine instead, if you have one non-sinusoidal process. So let's say you've got a sawtooth wave. A sawtooth wave that is, its fundamental frequency is 10 hertz, will have power at every integer harmonic. So it'll also have power at 20 hertz and 30 hertz in a decaying fashion. So you also have apparently many bumps. And a square wave, whose fundamental frequency is 10 hertz, will have power at every odd harmonic, 10 hertz, 30 hertz, 50 hertz, and so on. And so you cannot disambiguate or distinguish between if you have two different oscillations or one non-sinusoidal process from the power spectrum alone. So that's one of the reasons why we are not only doing this power spectral analysis with the FOOF package, but also this time domain analysis with this bicycle package. We do have an open issue right now on the FOOF package also to incorporate information from the phase spectrum to sort of bolster it even further. And so the idea is having all of these different packages and approaches be complementary and work together. So look at the phase spectrum, look at the power spectrum, and look at the time domain, and use all three of those to try and figure out what is the generating process that gives rise to this. But that's like a signal processing mechanism, but there's also just a physiologically plausible mechanisms, right? And so we also have to think about physiologically, how do neurons or neural systems interact? And actually I should expand that to say not just neural, but also glial too, like neuroglial interactions, because you get glial waves also. So they may be doing something as well. But we have to think about it's all complementary. You have the cool DSP analysis methods, but they have to be, in our cases, grounded in the physiology of what we think the possible generators are for these different signals. So I don't know if that answered your question. I hope it does, but since I can't see you, I'm just assuming you're smiling and like very pleased with that. And then I guess there's one more question, which is what is the difference between oscillations and cortical waves? So I assume you mean sort of like cortical traveling waves, which would be, there's a lot of really cool work like Lyle Mueller from like Terry Sinowski's lab here at UCSD has been doing some cool stuff on this too. Those would also be an oscillation. Those are just rather than like a point oscillation in a single time series, you have the spread of oscillations across the cortical surface. Whether those are being propagated by cortical processes or it's like one thalamocortical generator that just has different time delays. It's a little bit unclear. It's probably cortical is what I think the evidence is showing, but they're an oscillation that also is spreading through space. Okay, good. And when is my stop time? I just want to make sure that I'm not abusing time. I don't like it when speakers go over.
Usually go for an hour, and we started at 4:30.
Sounds good. So what's the solution to all this? My lab and I, we've talked ourselves into this corner of we don't even know what we're talking about oscillations anymore. So what might a solution be? So first we want to parameterize our time series or parameter, yeah. And so, this is an example of beta rhythm. Again, no, not a beta rhythm. This is 8 hertz theta rhythm, sorry. From rodent hippocampus. Rodents, I'm gonna like offend my animal colleagues, are like 80% hippocampal theta. They just got like really massive theta. Mice, when they're running around, it's so large amplitude, large of an amplitude oscillation. And so one of the ways that we're trying to quantify these non-sinusoidalities to see if that does something and matters is we sort of take that, we lightly filter it to try and emphasize the oscillatory component. And once we have this sort of signal, we can identify all the peaks and troughs and zero crossings, which is kind of funny. Like counting zero crossings is how identifying the frequency of a neural oscillation used to be done in like the 1950s. So I find it very funny that like we've gone back to finding zero crossings of our signal to try and identify oscillations now. But you can count these zero crossings to try and find individual cycles, and you can identify the peaks and troughs in those lightly filtered data. And then once you have that, you can then build an interpolated phase spectrum. So just sort of linearly move between 0, peak, 0, trough, and so on. And we don't do the zeros, technically we do halfway between peak and trough. But once we have done this and identified each zero crossing, peak, and trough of each individual cycle of an oscillation, you can then, for each individual cycle, calculate a bunch of different metrics. So you can say, how long did the peak last from one phase to another, and how long was the trough? What was the rise time? How many milliseconds did it take to go from peak to trough and trough to peak? How sharp is that? What's the asymmetry in that sharpness? And so for any given cycle, we can get the symmetry of the peak and the trough, the symmetry of the rise and decay, the individual amplitude of each cycle, the individual period of each cycle. And now for any individual cycle, we have a huge number of parameters. And so for people who are like in the Python world, the output of this is one big data frame where we identify each individual cycle, when each event happened, zero crossing peak, zero crossing trough, and then for any given cycle, these parameters. And once you have that really tight parameterization, you can then go on and do a bunch of other really fun analyses, which I'm not showing here due to the interest of time. And it's not technically published yet. It's still under review, but it's on bioRxiv, which is using hippocampal data from these mice and identifying each individual cycle. And this is open data from Yuri Bujaki's lab on the CRCNS. We can say these cycles or these oscillations aren't tonic. They come in bursts. The burst might be a 2-cycle oscillation. It might be a 20-cycle oscillation. But you have these bouts of oscillations that come in these like non-stationary bursts. And from these animals, Bujaki's lab also recorded individual putative excitatory and inhibitory neurons firing rates. And so we can do all these nice correlations between inhibitory and excitatory neural firing and these different non-sinusoidal features. But I think one of the coolest things from this paper that's on bioRxiv from Cole and I is you can actually, from these non-sinusoidal features of the very first cycle in a burst, predict how many cycles a burst will last for. We don't know why. That's just something that fell out of the analyses that's like really surprising, but kind of cool, which is whatever the non-sinusoidal features are of that first cycle, you can predict how many cycles that burst will last for. It's not a perfect prediction. It's not like 100%, but it's significant. It's better than chance. And you know, a couple of people in the lab are trying to chase down what that might mean. But that's a really neat kind of exciting idea. This idea of the sharpness of an individual waveform, you know, one of the first questions that might pop in your mind is, okay, well, where do these non-sinusoidalities come from? And so now I'm going to introduce you to this idea that what the local field potential is largely recording is not the outputs of individual neurons, but it's the postsynaptic currents and the transmembrane currents within that region. So it's largely the inputs coming into that region. And synaptic currents have a double exponential shape where they rise very quickly in time and then decay a little bit more slowly in time. And so this is based off of this like model of where sharpness come from is inspired by Stephanie Jones' work out at Brown, which is, you can imagine if you got a recording electrode in a region and you have a bunch of postsynaptic currents that have this double exponential shape. Here it's like an AMPA. Let's say you're getting a large excitatory drive input into a region and you're recording the postsynaptic, recording the LFP. If all of those come at relatively the same time with low temporal variance, those are going to integrate to look like what the initial kernel is, which is that one postsynaptic current that has this shape. Whereas if you have the exact same number of currents coming into that region, but with a little bit more temporal variance, those are going to integrate into a smoother individual waveform. So one argument about what is this non-sinusoidal, these sharp features of these waveforms indexing, it could be indexing, partially at least, the temporal variability of the inputs into that region, which is kind of neat, right? It's opening up some of the physiological interpretability of the signals that we otherwise would filter over and not pay any attention to. Not saying that is the answer. These are all like possibilities that we can start to interrogate using a variety of different approaches. So in addition to parameterizing our time series on a cycle-by-cycle basis, we're also working on parameterizing our spectra. And so this is work, again, largely done by Tom Donahue, and in the early days with my friends Matar Holler and Avgusa Shestuk, postdoc Eric Peterson, and sort of superstar undergrad from my lab at the time, Luanda Madanda. He's now up in Seattle living with you all. He's a machine learning researcher in Microsoft. I'm going to see if I can get him to come back to neuroscience someday. The package that we have that you can install and run, we released the official 1.0 a couple weeks ago, is basically does this. You feed it in a power spectrum, which is what this sort of, you can't see even here in black, the original data, and it decomposes that power spectrum into a background process, whatever that 1/f is it tries to fit, and then a sum of Gaussians atop that. So we try and do this initial fit to fit that background process, subtract it out, and then to those residuals, we try and fit as many Gaussians as possible, which we're calling the oscillations. I want to, and by the way, this works, I totally glossed over it. To get this to work on EEG, human, MEG, human, animal LFP from rodents, to get this to work pretty consistently across a number of species has taken years. It's like a very simple idea, fit this background process, subtract it out, and then fit Gaussians to whatever's left over until you hit the noise floor. Sounds very straightforward, very difficult in practice. And Tom did a huge amount of like software development work, making this work very nicely with these kinds of pretty output plots. So you can take a really complex neural time series, and with just a couple of parameters, you can capture a huge amount of the variance in that time, in that power spectrum, which are these 1/f parameters plus Gaussians that are defined by their center frequency, the power, the height above top of that background process, and then the bandwidth. And so I'm going to sort of end on this idea of, talked about the oscillations as the bumps. And by the way, we're fitting Gaussians, but you can fit any arbitrary sort of signal. We're digging into a little bit deeper as like, is a Gaussian the right thing, or should we be fitting like, you know, a gamma function or something like that? Gaussians are nice because the output parameters are easily interpretable. You've got where does the center frequency lie, which is the center frequency of your oscillation, and what's the height. So, you know, it's pretty nice, but there's this whole other aspect of that signal that we subtract out as stuff that we're not interested in, which is that aperiodic background 1/f-like process. So what is that? And I'm gonna sort of spill the beans here and say, this idea for me started from like a super simplistic and wrong idea, which is when I was in Adam Gasley's lab at UC San Francisco, I was interested in this idea of age-related cognitive decline. And reading the literature on that, there's this hypothesis, this neural noise hypothesis of age-related cognitive decline, which is roughly speaking in cog neuro sort of terms, as we get older, the mapping between the input that we get to our sensorium and the neural coding of that input just gets noisier. And so I was like, well, how do we, what is noise? How do we measure that? And I knew that the neural data that we're recording had this 1/f-like negative tilt in the power spectrum of the EEG and LFP. And so for people who don't come from a signal processing background, white noise, which is stochastic, pure randomness, has a flat power spectrum. It has equal power at all frequencies. And neural data has this correlational structure, which has low frequencies have higher power, exponentially higher power than the highest frequencies. And so I was like, well, maybe older adults, their power spectra look more like white noise, and so therefore are flatter. And so this paper in 2015 that we published was looking at basically that. That was a hypothesis I went in, that if older adults' brains are noisier, then their power spectra will look flatter. And that's what we found, is that older adults engaged in work-to-memory tasks have flatter power spectra, and there's this nice correlation between age and this 1/f noise. This paper has been replicated tons of times. This is like maybe the only time in my career that I can say this with confidence, that if you record EEG or MEG or ECOG from younger and older adults, you will see that the older adults have flatter power spectra than younger adults. Richard Gao from my lab said, I don't think that makes any sense. So, you know, as a PI, my job is to say, okay, prove it. Why do you think it doesn't make any sense? So I'm going to work you through this 2017 paper that we published that was trying to understand, where does that background 1/f-like signal come from? So I said most of what we're recording in the field potential are synaptic currents and transmembrane currents. If we want to be very simplistic, we can say there's AMPA excitatory currents and GABA inhibitory currents. Let's pretend that's all the brain has. AMPA has this very sharp like milliseconds rise, and then a couple of millisecond decay. And GABA has a similar rise, very quick, negative, right? It's inhibitory, but it's a little bit slower, and then it's a much slower decay. And if you simulate an LFP by taking a bunch of AMPA and GABA currents that are Poisson distributed in time, integrate those together, what you get is a simulated LFP here in black, which is you can decompose into the excitatory and inhibitory components. So if you just take an AMPA signal here, which has this double exponential form, exponential rise, exponential but slower decay, that signal in blue in the power spectrum will look like this. So it has equal power or relatively flat power out to a knee frequency, and it turns out to be around 70 hertz here, or log axis on the frequency, and then it starts to decay. GABA, because it's less sharp in time, it has equal-ish power out to its knee, which is only about 20 hertz, and then it decays even faster. And so as you change the ratio of AMPA inhibition, GABA inhibition, AMPA excitation, your power spectrum will move somewhere around in this space. And so the argument that we put forward in this is that this background aperiodic signal is capturing the relative amount of excitation and inhibition to that region. There's other transmembrane currents, of course, there's other neuromodulatory currents. NMDA has a very long time scale as an excitatory, but simplistically, we can say that this background aperiodic shape is capturing some amount of, and we called it the slope of this background, is capturing some amount of this excitation inhibition balance. We tested this in a variety of different ways. This is kind of like the lab MO. If we have an idea, we can run a very careful experiment that takes many years and costs a lot of money, or we can find if somebody has data that's pretty close to what we want and see if we just analyze those data instead. And so we looked at some data from the NeuroTycho database, which is Cortical ECOG in macaques. And in that database, they have macaques that are under propofol anesthesia. Propofol is a heavy GABAergic anesthetic. So we knew that if our model was correct, we should see that at rest, there should be some slope of this power spectrum, which happens to be at around -3, you could see down here. They inject the propofol into the monkeys, the power spectrum goes a little bit wild. And then once they're in this anesthetized state, the entire power spectrum shifts to a -3.5 to 4 state, which is what our model would predict. If you go from less excitation to more inhibition, you'll go from less of an AMPA state to more of a negatively sloped GABA state. And so it stays in that until the animal begins to recover out of the anesthetized state, and then it flattens out. And this happens cortex wide, except for this little bit where it's in the opposite direction in the posterior cingulate sort of area, which is kind of fascinating, and don't know why, don't know what that means. But otherwise, broadly cortical, it's this negative slope effect. And we had a couple of other tests in that paper using other data sets to try and push at this. And so this was exciting to us because this like EI imbalance kind of idea is prevalent now in like everything. Everybody's super excited about excitation, inhibition balance. But it's in really, you just can't measure it in humans in a dynamic way. You can try and get estimates of excitation, or I should say GABA and glutamate, using MRS, magnetic resonance spectroscopy, but that's static in time. And these are, what we're arguing is that if this method is correct, and this is what the 1/f like slope is capturing, now we can get a dynamic measure of relative excitation inhibition in humans in a way that we otherwise wouldn't be able to do. And there's now a ton of follow-up studies. People have found DBS, not DBD, DBS and depression. I should actually say I'm a co-author on some of these, so it's not entirely fair, or unbiased, I should say. You know, a number of people have found now these aperiodic slope effects in a variety of different conditions, aging, development, DBS and depression, schizophrenia, sleep, and so on. There's lots of really cool new stuff happening from a variety of different labs in this area. Okay, so I'm gonna end on this. With parameterization, once we have this ability to do fine scale parameterization, turning our complex neural data into a series of numbers, and this is like the love of data science here, we can then run large scale analyses that were otherwise not possible without doing so, such as, and I'm gonna end on this paper that we posted by our archive yesterday, integrating a ton of different open data sets. And so I'm gonna give a shout out to John Murray here, who had this cool paper in Nature Neuroscience in 2014, where he took data from a bunch of different single unit macaque recording labs and took all those different data sets and said, okay, let's look at single unit spiking statistics in all these different regions from these different groups. And we're going to look at them as a function of the anatomical hierarchy. And so the anatomical hierarchy, the idea is that the anterior-most parts of, say, the frontal cortex represent very long timescales information, right? It's like goal planning and decision-making and things like that. And this is kind of inspired some on the cog neuro side by like David Botter and a couple of other folks, which is then as you progressively move from anterior frontal cortex back to posterior, you go from very long-term goal planning and decision-making to DLPFC, which is more short-term, like working memory maintenance, back to supplementary and premotor and motor areas, which are like more immediate stimulus response kind of areas. So you have this sort of hierarchy of temporal processing along the frontal cortex, and it turns out there's many of these different sort of hierarchies of processing. And the way that Murray in this paper quantified these temporal processing timescales was by looking at the intrinsic timescales of the single units. And so an intrinsic timescale is a little bit hard to explain in the last couple of minutes I have, but if you take a signal like a neuron spiking statistics and you imagine a very simple stimulus response unit in like V1, that unit will fire an action potential, a number of action potentials very quickly in response to an input, and then stop firing once that input goes away. It has no memory of that input over time. And so if you look at that spike, not no memory, but very little. And if you look at that unit and you say, okay, how well can I predict its spiking patterns right now from data 10 milliseconds from now, 20 milliseconds from now, 40 milliseconds from now, whatever. That ability to predict that autocorrelation of that signal drops very quickly. So a unit in primary motor cortex, primary auditory cortex, primary visual cortex, its autocorrelation drops very quickly. You cannot get any information or very little information about what the spiking looks like right now from how it's going to look, 10 seconds from now. Whereas if you look at a unit in like DLPFC, lateral frontal cortex, ACC, there is a higher autocorrelation. The spiking looks more similar over time. And the rate at which that spiking decays is the time scale of that neuron. And John Murray showed that as you move up this cortical hierarchy from visual areas all the way up to anterior cingulate cortex, the time scale of these neurons gets longer and longer and longer and longer, which is pretty cool. That's really neat. And so we thought that's really neat, but we can't do that in humans. But we can get an estimate of the population time scale from the ECOG or the field potential signal in these people. And so I'm actually going to skip this slide because we're running out of time. But if you take different time series, these are simulated to have fast time scales, medium time scales, slow time scales. So this is what the autocorrelation looks like. The black signal, the signal is very correlated to itself at 1, 2, 3 millisecond lags. But once you're out to about a 20 millisecond lag, the signal with itself, the correlation drops to almost 0. Whereas in this signal here in yellow, it has a higher autocorrelation, and you can quantify the time scale as this decay time constant. The problem is oscillations in the signal really mess up your ability to define time constant as a function of its autocorrelation. But it just so happens that when we were writing this FOOF algorithm, we fit the algorithm such that it fit this sort of knee function here. So it's not just fitting a negative slope, it's actually fitting this kind of function here. And mathematically, Richard Dow was showing that this location of the knee is the time constant of the neural population, this field potential. Actually, it's proportional to it. The time constant technically is 1/2 times pi times the frequency at which this knee occurs. And so in simulation, we show that we can simulate data with different time constants and then recover them from that FOOF algorithm that I was showing, which is here's where we find the knee in the FOOF algorithm in the data, and it sort of very nicely matches the ground truth in simulation. And so then we took that neurotyco database that I showed you, where it had the macaque ECOG signals, and said, okay, well, let's take John Murray's single unit spiking time constants in all of these different regions from that paper, and take those same regions in the ECOG from another set of monkeys in that neurotycho database and try and fit our time constants to the ECOG and see do they match up. And you can see that the ECOG timescales that we're inferring from a different set of monkeys in the same region correlate with the single unit timescales of a different set of monkeys from different papers, but in those same regions. So this is showing that we can get this measure of the timescale of a neural population from the field potential by using this approach. And there's a lot of work going on right now about these like timescales of neural populations where you have like I said, these like lower sensory and motor regions have these very rapid timescales. And as you get higher up along the hierarchy, the timescales get longer and longer and longer. And that may have something to do with the ability of the neural population to encode information over longer time periods. Right? You want a visual system to respond very quickly and then forget that information and pass it along for further processing. Whereas you want, you know, other systems to be able to hold information for very long time periods, but you don't want those two to be the same thing. And it turns out that you have the cortex has these nice gradients where it seems like these timescales sort of increase as you go across. And so from another database of approximately 106, I think it's exactly 106 people who have these ECOG grids implanted, we can take a look at their timescales across the entire cortex and take another database from the Human Connectome project and look at the amount of myelination. And you can see if your timescales, the map of your timescales, follows the amount of myelination. So early cortical, early sensory areas and motor areas, so here you can see visual cortex, have dramatically different myelination patterns than like association cortical regions. And we find that as you move from sensory cortices, where we have very fast timescales as inferred from these ECOG signals, up to association, the timescales begin to slow along this myelination gradient across the human cortex. So we're pretty excited about this because this method of inferring timescale from the ECOG signal means we can also do it in a task-related way. So we took another database from Robert Knight's group. This is from Liz Johnson of patients, 17 patients doing a working memory task, and said, if these timescales are somehow related to how long the neurons are holding information in time, then we were curious as to, are these timescales static and a result of purely the structure of the cortical region, the myelination, or are they dynamic? Can they dynamically change in response to tasks? And so we find is that if we look at the power spectra at pre-stimulus baseline and power spectra during these delay periods and fit these timescales to the different time periods, that within the parietal cortex, prefrontal cortex, orbital frontal cortex, and MTL, where we had electrodes, timescales always increase ubiquitously from baseline to these working memory delay periods in the 4 regions that we have data from. So the delay period baseline timescale, I should say, as a function of baseline, it's always greater than 100%. But specifically in the prefrontal cortex, the change in timescales from baseline to delay period actually predicts individual working memory performance on this task. And so these are a ton of different data sets that we tried to bring together to understand, you know, what are these timescales potentially doing? What are they structurally related to? And are they functional? Are they doing something? And so we're pretty excited about this one. You know, we wrote this code when we first wrote the FOOF algorithm. We fit this need because we knew it was in our data, but we just didn't know what to do with it. We're like, well, you know, it's a parameter that we have to fit to our data to make the fitting work. And then, you know, 2 years after writing that, we now have this, well, maybe that parameter is actually physiologically interesting and we could potentially do something with it. So that's been pretty exciting. Anyway, I'm going to stop. That's a lot of stuff. I don't want to keep going. I have a lot of like big ideas about where data science should interface with neuroscience, but one of them is there's a huge amount of really high quality data out there that we can be leveraging to start to address really fundamental questions in neuroscience. And so I'm a big fan of running the experiment that I want to run. I have developed tasks that I've, you know, had to pilot on hundreds of people to make sure that my task works. And sometimes you have to develop that task if you want to ask the exact question you want to ask. But sometimes you can look for the data that's already out there and get like 80 to 90% of the way. It's not the perfect data set, but it's really close. And we've got a bunch of those 80 to 90% of the way there data sets, and you analyze them all. There's something very nice to me scientifically about using other people's data where there could have been no bias in the collection to try and prove a point, and see if those data help actually hold up to the theories and arguments that you're putting forth. So anyway, that's it.
Thank you. I'm sorry you can't hear everyone clapping. Okay.
Oh, it's okay.
I'm
used to this after my talk, so everybody's being like, oh my God, just silence. Why did I do this?
Okay, so we have several questions that have been posted. I'm going to call on people. I'm going to unmute people, and hopefully you can ask your own questions just to make this a little more interesting. We're going to go with Michael Nolan, followed by Noah Benson. Okay, so Michael, I'm going to unmute you, hopefully, and you can ask your own question.
Okay, cool. Can you hear me?
Yes.
Yep.
Great. Well, first of all, thank you for the talk. That was fantastic. My question is kind of a few points back, though. Like you said, you covered a lot. But this idea of excitatory inhibitory imbalance having an effect on the overall sort of tilt of kind of the 1/f background noise. And I was wondering, relating that to some of the stuff you were showing at the very beginning of the talk with these cortical or even just general cerebral organoid models. Like, I'm not super familiar with like different interventions you can have in development, like say to suppress or amplify like the relative, just to affect that balance essentially, to see like if you can screw this up, does the spectrum produced from these organoids kind of reflect what you're expecting? I mean, you know, not to assume the experiment worked before you do it, but whether or not that work's been done, whether you think it can be done, and also this sort of, sorry, this is a long question. This like super highly coordinated activity that you're seeing with sort of field potentials not really having the high complexity that you see in more full cerebral models. Like, is that like a transient thing or?
Yeah, so we are doing those experiments in the organoids too. So not only can we, you know, one of the wild things about the organoids is we have full genetic and optogenetic control. So we do optogenetic manipulations in them potentially, not quite yet as far as I know, but you can do some genetic manipulations, knock-in, knockout, for example, to alter the development. But even more simply, you could just do pharmacological manipulations too, right? And so that is work that we are doing. There's a really cool paper from a number of years ago in Neuron by Flavio Froelich as the first author, showing that in vitro slice work, applying very low amplitude, sub-threshold oscillatory electrical fields, biasing spiking in the slices. And so Richard Gao from my lab is actually doing that in the organoids. So embedding the organoids in very low amplitude electrical fields to see if that alters the spiking statistics within the organoid. And we can also do some of the EI balance work too. The other side of things is testing some of these theories of EI imbalances in like different kinds of disorders of growing cortical organoids from populations with genetic disorders that are hypothesized to have an EI imbalance component, and then looking at that in the organoid compared to like control organoids. So yeah, that's work that is going on. I am not the lead, so I don't get to determine exactly what studies are being done. I'm at the whim of my collaborator, Alison Motry, but we have a lot of really cool sort of fun ideas that we're poking around at.
Great, thank you.
Yeah, you're welcome, thank you.